my name is Miha. I work for a Cambridge-based biotech company called Kymab. And what I wanted to talk to you about today is how we're using the BD Rhapsody platform in our translational immunophenotyping. We did single-cell RNA sequencing using the BD Rhapsody platform, and I'm going to tell you a bit more about that. And then we also did bulk RNA profiling using the nanostring. In terms of sort of the experimental part for the on the BD Rhapsody, we loaded a single sample per cartridge. We encapsulated them with the beads. We also used the sample multiplexing tags. Basically, it was a bit overkill, but it gave us extra information. Then we pulled out the beads, generated the libraries, sequenced the libraries, and then we used the computational workflow that is captured in the 7 Bridges platform, but we also used sort of our more bespoke computational pipelines to tidy the data up and for data visualization. To give you some numbers, so like I said, we had 5 donors. We had paired PBMC and tumor samples, so 10 samples in total. We obtained, so after the computation, the sequencing and the early computational workflow, we got just shy of about 80,000 cells for these. After the more sort of bespoke computational cleanup, we were left with, I have to say, we used the immune response panel, like the previous speakers. Obviously, we're interested in immune cells. We use the human immune response panel. So after the cleanup, we had expression for 366 genes from just over 63,000 cells. And as you can see, so from the table here, each library is basically a single sample, and you can even see the annotations. So these are PBMCs, tumors, and so on. We had approximately equal representation of cells for each sample, with the exception of 2 tumor samples, this one and this one, where we had a larger number of cells, which is mainly due to an increased number of tumor cells that actually got loaded on the cartridge. So this is a principal component analysis plot of all of these samples after the filtration of cells, the normalization, and the batch correction was done. You can see that basically the samples roughly sit on top of each other, which just is a good QC to tell us that we've removed sort of the major batch effects, and that what we're dealing with in terms of the differences in gene expression is due to the biology of these samples. But obviously also one thing you can see from the PCA plots is that you can't really see differences in cell types. So for that you need to turn to different computational approaches. So what we've been doing is we've used UMAP. UMAP is it's very similar to TISNE. It's a dimensionality reduction algorithm, but it's got some performance benefits over TISNE. So if you can imagine that on a data set of over 60,000 cells, TISNE does take quite a while, whereas UMAP goes through these in maybe 30 seconds, maybe a minute on a standard desktop computer. So it's just a very efficient algorithm, but essentially in terms of what we're visualizing here, it's very similar to TISNE. So these are sort of the two-dimensional projections that we get from the PBMCs. And if we look at the sort of the classical marker genes here, so we've got a color gradient to basically just visualize the expression patterns. What we see is that essentially we've got a huge cluster of T cells, we've got a cluster of B cells, we've got a cluster of myeloid cells, so this is showing CD14 expression, so these are your classical monocytes. And then if we drill down a bit more, so in that T cell compartment, we've obviously got CD4 cells, we've got CD8 cells, And then something that obviously the marker here is called Fc gamma receptor 3A, also known as CD16. It's expressed on two very different types of cells. So on the one hand, we know that non-classical monocytes express CD16, but also NK and NKT cells. And we can see that basically these cluster closer to CD8s, which is expected, and these guys cluster closer to the classical monocytes. So this sort of from a top-level view all looks like what you would expect. Obviously, if you look at the tumor samples, there's a lot more, just looking at the sort of the structure of the data, there's a lot more going on. And if we look at, again, sort of these standard markers, we've got CD3, so we've got T cells, but you can already see that there are differences in the structure of the data. We've also got B cells, so these are, if you recall, these are non-small cell lung cancers. There is quite an sizable component of B cells in lungs in general and in lung tumors. Again, we've got the myeloid cell component. So these cells express CD14, so they'll be your classical monocytes or your classically derived macrophages. If we drill down again a bit more, we've got CD4 cells here. We've got CD8 cells. And then I just basically looked at a marker that is known to be expressed in lung epithelial cells and is known to be upregulated in tumor cells from lung epithelial cells. So that's CD24. And we see that we've got quite a few different clusters of these epithelial stroke tumor cells. And I can tell you that these clusters are mainly, they differ based on the donor they came from. So this is known in the field that you have more of a universal immune cell compartment from different donors, whereas there are differences in the tumor cells from the different donors. So obviously, again, from a top level, this looks like what you would expect from these samples, but obviously using the BD Rhapsody, we're not just measuring these 6 markers. We're actually essentially measuring 366 markers. So there's a lot more information that we can actually get from these samples. So I've looked at various different computational approaches to get as much information out as possible, and we had a conversation at lunchtime about this. So like Ricardo said at the beginning, you can see the structure in the data, but it's actually quite difficult to find discrete clusters of cells. So we actually opted for a different approach, sort of looking at it from the other end, i.e. we've assembled gene expression signatures for 27 different cell types. So we tried to be as broad and as comprehensive as possible. And looking at those gene expression profiles, what we've done is we've used an algorithm that then scores the actual gene expression for each cell out of the 63-odd thousand that we had, and looks for whether it's most likely to be a B cell or a T cell, and it actually then stratifies each individual cell into these 27 bins based on the expression profile. So what that enabled us to do was look at, so on the one hand, all of these cell types, we could look at which ones were present in our data set, but also what their functional properties were. So if you remember, obviously this work was done for a specific antibody program. So initially what we wanted to do was we wanted to see if the expression patterns of ICOS on these different immune cell types mirror what we were seeing in preclinical models and what was already published in the literature. So essentially, we pulled together those different cell types into 4 bins that we were mostly interested in. So those would be your Tregs in brown, non-Treg CD4 in blue, CD8s in green, and everything else in yellow. And we wanted to look at the ICOS expression levels both in the tumors and in PBMCs. And what you can see is that So obviously, cells that don't express ICOS have a large peak that's negative, both in the tumors and in the periphery. But what we can clearly see here is that ICOS expression is highest in Tregs, specifically in the tumor microenvironment. It's a lot lower in Tregs in the periphery. And obviously, if you kind of consider back, I was saying that we want this dual mode of action. So we want to be depleting the cell type that is responsible for immune suppression in cancers, but we don't want to be depleting cell types that can actually clear the tumor cells. And that is very much dependent on target expression on these cell types. So this is showing exactly what we were already seeing in preclinical models. And it's giving us confidence that basically the highest expression of ICOS is actually on cell types that we do want to deplete, and the cell types that we want to engage with in terms of the co-stimulatory part of this dual mode of action are expressing less, but are expressing enough for us to be able to engage with the target. This is also sort of captured in this table that's color-coded. So this is ICOS expression in specifically intratumoral T cells. So the cell types are ranked based on the percentage of ICOS expression, so the highest here in blue, the lowest in red. So it's basically just kind of summarizing what I've said here. So the highest expression is in Tregs, so just over 81% of Tregs also express ICOS, followed by activated CD4 cells. At the other end of the spectrum, the lowest expression is found on NKT cells and CD8 effector cells. What we can also do is enumerate or look at the fractions of these cells, and that's sort of highlighted here again in a color code. So the most abundant cell types that we found in these samples are in green, and the least abundant are in brownish yellow. So we see that we've got about 1.4% of Tregs in the tumors, which is kind of what we were seeing with our FACS data. For example, we can see that the most abundant cell type in the T cell compartment are actually resident memory T cells, roughly 19%. The least abundant, for example, are Th17 cells, just over 0.1% of the cells. So these are all the things that we can then sort of glean from the data just by doing single cell RNA sequencing. We get a lot of information essentially for free. So, in summary, we've basically successfully used the BD Rhapsody platform for in-depth immunophenotyping of cancer patient samples. Essentially, this was mainly done to support the preclinical work for our KY1044 program, and so obviously primarily we had an interest in ICOS expression in different cell types, but also this provided a link between FACS data and bulk RNA data, and also it gave us a good indication of that what we were seeing in preclinical models was also kind of being mirrored in actual human patient samples. And I just wanted to kind of finish off with some technical observations from this piece of work. So one thing that we've noticed was that certain genes are just not being picked up. And as, working in the field of immuno-oncology, PD-1 is an extremely important gene. So I'm not sure if you are aware, but Tasuke Honjo won the Nobel Award last year for its discovery, so for the discovery of PD-1 and its use in immuno-oncology. So for us, it's probably the most important gene. However, we've found less than 10 copies of this gene in over 120,000 sequenced single cells. And from FACS data, we know that it's expressed, obviously it depends on the cell type, but it's expressed to a quite appreciative level. So the other thing that I found interesting, and I've noticed so it's well known, but I think it doesn't hurt to reiterate this, is that CD4 expression is not not actually exclusively seen in T cells. So there's a considerable level of CD4 gene expression in myeloid cells, particularly in DCs. Obviously, in FACS, these DCs are then dim, not negative, but dim for CD4. So there are obviously post-transcriptional or post-transcriptional regulatory signals after the mRNA is actually synthesized. But obviously, from our point of view, I think as sort of a community, we need a good catalog of which mRNAs are being expressed in which cell types. And that might not actually be the same marker genes that we're seeing expressed at the protein level. One thing that's an important point for me is that we're picking up only about 10% of granulocytes. So only about 10% of all the classified cells are classified as granulocytes, even though we would expect a lot more. So we know that neutrophils are the most abundant cell type in blood. The reason, initially I thought that the main reason is the tissue processing. So we know that neutrophils are a very, very difficult cell type to work with. They burst and die if you just look at them funny. But I've recently kind of started thinking that it's actually more likely that they're being filtered out because they don't express a huge number of genes to a very high level. So it's their transcriptional quiescence that's actually meant that we're filtering them out because they resemble more cells that are either dying or that cell types that aren't being picked up by the immune response panel. So we're only picking up a fraction of the granulocytes that are actually present in the sample. I think that AbSeq, so unfortunately this work was done before AbSeq was launched, and I think that AbSeq will help with all of these points that I've raised because it just adds the protein part to the missing sort of signals or information that we're just not getting from the mRNA. So I think it's a really great technology. It was one of the main reasons why I thought that the BD Rhapsody system is what we should be going for. I'm very keen to get my hands on it, and I think it will just really help with these issues. And with that, I really need to acknowledge everyone that's worked with me over the last couple of years on especially the KY1044 program. You can imagine that it takes a lot of very highly skilled people to generate an antibody and get it to the clinic. So the clinical trial is up and running.